is Ethan from Laura Harrington's lab at Cornell. Thanks.
All right. Good morning, everybody. And thank you, Eric and Maurice, for the opportunity to present. My name's not up there yet. Oh, there we go. Okay. My name's Ethan Degner. I'm a student of Laura Harrington's at Cornell University. And I'm going to be switching gears a bit. And I'm going to be talking about mosquito sperm. And this is maybe something you don't think about all that frequently, but I'm here to argue that maybe you should, because sperm are just as important as the more familiar female egg at producing that next generation of pathogen-transmitting mosquitoes. And they're directly related to a male's reproductive success. And so this is going to be an important consideration for strategies, control strategies that use mass releases of males, such as sterile insect technique. There are aspects of mosquito sperm biology that we don't really understand very well, but are going to be important. And to give you an example of this, cytoplasmic incompatibility in Wolbachia-infected Aedes aegypti, we don't have those mechanisms resolved yet, but it's likely that sperm are mediating the incompatibility between the Wolbachia-infected male's sperm and the uninfected female's egg. And then, as Doug gave us a great introduction to, sex is important and could be a valuable target, and there are likely vital reproductive processes surrounding sperm and within sperm that could be potential targets for future vector control strategies. So with this talk, it's my goal to convince you that this is both an interesting and an important system to study. I'll be taking you on the same journey that sperm take through the female's reproductive tract to their ultimate destination of the egg. And I'll be highlighting along the way questions that we still haven't resolved as a scientific community. Ultimately, I'll describe a method and a resource that I've been developing so that we can understand these cells a little better. So if you've never seen mosquito insemination, here's your chance. So this male's going to clasp the female's terminalia, and she's going to extend her final tergite, and then the sternite's going to open like a trap door, and you just saw intromission. So I apologize if that's a bit graphic, but this is what it looks like, and from here on out, the male's done. We're not going to talk about him anymore, and we're going to focus on the female's reproductive tract. I want you to become familiar with this anatomy because I'll be coming back to it for the rest of this talk. So a female's site of insemination is her bursa. It receives both semen and sperm. The long-term sperm storage organs are spermathecae, and in culicines there are 3 of them. And then the common oviduct runs ventral to everything. And the bursa and the spermathecae are connected by spermathecal ducts. And I want to take this moment to highlight that I'm using Aedes aegypti as a model. So that's a culicine mosquito. But the anopheline mosquitoes, our malaria vectors, are going to have a bit different morphology. So they won't have this bursa, and they've only got 1 spermatheca. So everything I say here, or not everything I say here, may be generalizable to all mosquitoes. So zooming in on this reproductive tract, sperm are briefly held within the bursa, and they're dispersed throughout the bursa. But very quickly, they end up getting activated within that bursa to a pretty rapid motility. And Catherine Thaler and her colleagues at UC Riverside have described this pretty eloquently in Culex quinquefasciatus. So sperm are initially dormant, but serine proteases within the semen activate those sperm. And this motility is dependent upon the extracellular concentration of calcium, and it's also powered by kinases within the sperm. So they'll undergo a few different sequential progressions through different flagellar waveforms, and ultimately they'll end up in a rapid helical waveform that you should be able to see here. Not really, sorry, this lighting's not very good. You'll have to look at their paper, but this video was by Thaler et al. And so they did a great job of characterizing sperm motility in vitro. In vivo, these active sperm are going to be congregating near where they need to be stored by the spermathecal ducts. And this is a pretty rapid and almost violent motion. And I've taken video of this. Here's the bursa and the spermathecae. And if you look closely, you can see this mass of sperm in between the two, moving the wall of the bursa. And Thankfully, this female was inseminated by a male that has fluorescently labeled sperm. And so if we flick off the lights in a second or two, you'll see this mass of sperm. And it seems like it's moving almost in concert or in a coordinated motility. So there's about 1,000 sperm here, and they're all trying to get stored. So very soon after insemination, those sperm will begin to store, and they'll ascend the spermathecal ducts and end up in the spermathecae. After about 5 minutes, sperm storage is complete, and there, sperm will wait for a female's entire life for the opportunity to fertilize an egg. When that time comes, days or even weeks after insemination, a female will release 1 or 2 sperm with a descending egg, and one of those lucky sperm will enter the egg through a tiny little hole in the back of the egg called the micropyle, and only then is fertilization complete. So this path has been fairly well described using microscopy, but there's still so much that we don't know about it, and especially on the molecular level. So I want to highlight a few of those unknowns. I mentioned that what activates and powers motility has been described, but in the medically important vectors, we don't really know the proteins that are on sperm that are involved in regulating this motility. And we certainly don't know the conditions within a female that allow sperm to navigate her reproductive tract. As I just mentioned, we don't know how they're getting to where they need to be, and they could be following a chemical gradient. They could be using chemotaxis, or there may be some sort of physical structure, but again, this is kind of a black box. Once sperm begin to store, it would seem logical that they could just swim up themselves, but other authors have proposed that females actually create a negative pressure gradient in the spermathecae that sort of sucks sperm in. And I think that both mechanisms are involved, but we still don't fully understand this. And so I'm going to talk about that later in the talk. And then, how does a female maintain these live, pretty much helpless cells for weeks? She must be nourishing them. And this is an interesting question, though, because other investigators have proposed that these sperm don't actually have functioning mitochondria, or at least functioning electron transport chains. So it seems like they may not be able to really provide themselves with sustenance. And work in Flaminia's lab has shown us that the spermathecae provide oxidative stress relief to these sperm, but we don't fully understand all of the proteins, especially within the sperm, that would provide this service. Ultimately, when these sperm are released, they'll swim back down the spermathecal duct and enter that egg, but how do they actually fuse with the egg and recognize it? We still don't understand this. So that was a lot of questions, and these are the kind of things that keep me up at night. And as I've been pondering, yes, I'm a bit weird. As I've been pondering how to begin to tackle this system, one thing has become clear, and that's that there isn't much of an investigative infrastructure surrounding this system. And so it's been my goal to develop a framework by which I and other investigators can begin to understand how sperm function within a female. So I'll be describing a resource and a tool that I've been developing to do this. Again, looking back at those questions, I realized that the answers to a lot of them were proteins. And so I delved into the literature to find all of the proteins that I could that had been described in mosquito sperm, and there's been a few at most. And so my goal then, or what I've been doing, is using a top-down approach and using shotgun proteomics to identify all of the proteins I can within mosquito sperm as a first step. So I dissect out the male accessory gland, the male reproductive tract, and I get rid of the accessory glands because those contain the semen, and we already have that proteome characterized, so we don't really need to know what's in there anymore. But the seminal vesicle, which is what's still highlighted here, contains the mature sperm, and that's what I want to identify the proteins in. So I cut the tip off, and then with a minution pin, I'm with very careful hands, I'm able to squeeze out the sperm like a tube of toothpaste into a cloud. And multiply that by 800, and we had enough sample to run mass spectrometry on these mosquito sperm. And so I have to be honest, this data is fairly hot off the press. We just got it towards the end of last week, so we haven't fully curated it yet. But we do have roughly 1,000 proteins confidently identified within these mosquito sperm. And looking at that list of proteins, I want to highlight a few that we think might be interesting as candidates for future analysis. So I mentioned that ion channels in the sperm and kinases are responsible for regulating motility. So we'd like to identify the specific proteins within sperm that do just that. We found a good number of DNA binding proteins like protamines and histones, and we expected this, but now that we know these accession numbers and where they're located in the genome, we hope to develop a transgenic line that has the sperm head fluorescence so that we can very easily track how sperm move through a female reproductive tract or understand how different male sperm may compete within a female reproductive tract in the case of a polyandrous female. And then there was an odor binding protein or two, and we thought that this might be a prime candidate for studying whether females or whether sperm actually are using chemotaxis to traverse the female reproductive tract. And then with all of these proteins, we hope to use CRISPR to knock some of them out and then characterize their reproductive phenotypes using these mutants to test their loss of function phenotypes. And we have this CRISPR system working in our lab with Aedes aegypti, and so we're hopeful that that will be up and running soon with these proteins. So the next thing that I've been developing is a technique to introduce just about anything we want into the female reproductive tract. We wanted to be able to introduce things like inhibitors for some of these proteins or maybe chelators of calcium. We're running out of time?
No, we're good.
Okay. Chelators of calcium that might be affecting sperm motility. And so I've developed an artificial insemination technique with Aedes aegypti. And I'll run briefly through this in case you're interested in using it. I harness this female very carefully with a cut micropipette tip. And then using micromanipulators and a forceps, I'm able to very carefully pull on her cerci to expose her site of insemination, the gonotreme right here. And with a microinjection needle, I'm able to deliver 3 or 4 nanoliters of just about anything I want. And in this case, I've used red food coloring as a proof of principle. So you can see her bursa is very neatly filled with this red food coloring. But when I introduced fluorescent microbeads in with this dye, I noticed something interesting, and that's that those fluorescent beads are initially contained within the bursa, but sometimes they also end up stored in the spermatheca. And this intrigued me because it doesn't happen immediately. And so it seems like a female may be actually actively storing inert seminal components from the bursa into her spermatheca. And so maybe she's even storing things like seminal proteins or steroid hormones or anything like that in the spermatheca that could modulate sperm function after they've been stored. We know that this happens in Drosophila melanogaster, in which sex peptide binds to sperm and then rides them into storage, but controls the release of sperm from storage long after a female's mated. So we're interested in exploring this further, and we plan on using our sperm proteome and our semen proteome to do mass spectrometry on male-derived proteins within the spermatheca to understand what proteins might actually be getting in there. The other thing that we glean from this experiment is that since inert components are actually making it to the spermatheca, it may actually be true that females are creating a negative pressure and assisting sperm into storage. So in conclusion, I've described a system to you that I hope at least some of you weren't already familiar with, and I think it's a really important one to study. I've also described a framework by which I hope that I and other investigators can begin to unravel the mysteries behind these mosquito sperm and how they function in a female. And it's my hope that ultimately some of this work will lead to improved or new vector control strategies. So with that, I thank my advisor, Laura Harrington, all of our Harrington lab so beautifully dressed as Muppets for this Halloween, and our proteomics collaborator, Steve Doris at Syracuse University. And thank you for your attention, and I'll take any questions.
Flamini. She has a
Oh,
question there.
Flamini. Yeah. Thank you.
Do you know, can you speculate on what's the role of female proteins in sperm storage? And also, I'm interested in what you said, that you think that like during release of sperm for fertilization, you get like 1 or 2 sperm out of the spermatheca. So do you know how that is regulated? I mean, in Anopheles, we think it's actually quite a few
Oh, excellent.
sperm cells that are released every fertilization cycle. So
Okay.
just curious to know why you said
I'm going to address the last question first because that's the one I remember. So the question was why we think only 1 or 2 sperm are being released. So that work was done, I think, in QLEX, and it was done in the 60s. But I have to be honest, their methods may not have been the most rigorous. So it could be that more are being released. There's been some work that has counted the number of sperm in the spermatheca and then tried to correlate it with the number of eggs that have been produced, and that work wasn't really done quantitatively rigorously, I don't think. So I guess I'm not fully sure how many sperm are being released, but it probably is limited. And what was your second question?
So the first question.
The first question.
Was whether you think, what do you think the role of female proteins or female factors is in sperm storage and sperm survival?
Absolutely. Are you talking about like spermathecal secretory proteins? Yeah, so we know from your lab that the spermathecal secretory gland is producing things like heme peroxidase and other things that will protect the sperm from oxidative stress. Other insect sperm literature has claimed that glycoproteins and lipoproteins will be providing sperm with sustenance. As far as how they might be metabolically maintaining these sperm, I'm not really sure at this point. Yes?
You said that on occasion you sometimes see fluorescent beta you're cleverly injected.
Yeah.
Making it into the spermatheca. Why do you think it's sometimes?
So I'm actually not sure about that, and I plan to look into this further. There is a correlation between time post-mating and how frequently the spermatheca are actually filled. So if you wait a day or two, most of the time the spermatheca will actually be filled. And I don't know if, so this bursa isn't musculature, or isn't muscular, it doesn't have muscle surrounding it. And so it's unlikely that they're actually pumping things in, but I've wondered if when females take a blood meal or take sugar meals, if that kind of pushes things from the bursa into the spermatheca. That's a good question.
Yes, just a curiosity, a general question. There is any correlation that EDIS could have an increased polyandry with respect to an ovaries, as an example, because of the fact that you have 3 spermatozoa instead of 1? Why do they have 3 instead of 1?
That's an excellent question. So nobody really knows. That's the short answer. So I did some polyandry work, and I was able to look at those red fluorescent sperm and the white sperm, or the wild type sperm in the spermatheca. And that's what I was hoping to see. I was hoping to see that there would be differential storage of sperm and that she could choose. And there didn't seem to be really much of a pattern to which sperm got stored where. And so why they have 3 spermatheca and why they usually don't use one of them is still kind of a mystery. And if you have theories, I'm happy to hear them. But yeah. Okay. Thank you guys for your attention.